Hi, have you ever wondered what gives orange juice its signature tangy smell? That dominant tart flavor comes directly from citric acid, the primary organic acid found in citrus fruits. In the food and beverage industry, measuring this acidity is just about taste. It is a crucial step to guarantee strict quality control, nutritional standards, and to ensure a consistent flavor profile in every single batch. In the chemistry lab, we quantify this using a standard method called acid-base titration. During this procedure, the orange juice sample acts as our acid and is titrated against a standardized sodium hydroxide solution. By utilizing a phenolphthalein indicator, we look for the exact moment the solution turns a persistent faint pink, signaling that complete neutralization has been achieved at the endpoint.
Now I will explain about the theory of the application. Citric acid is a weak three-protein organic acid that is naturally present in citrus fruit, especially orange juice. The concentration of citric acid in orange juice is determined using an acid-base titration method. In this experiment, sodium hydroxide, a strong base, is used to neutralize the citric acid present in the sample. At the endpoint, the moles of sodium hydroxide used are stoichiometrically equivalent to the moles of citric acid present in the orange juice. This relationship is used to calculate the concentration of citric acid in the sample.
Now, let's look at the methodology used to determine the citric acid content in orange juice using an acid-base titration. First, the burette was filled with 0.050 molar of sodium hydroxide or NaO, which acted as the titrant, and the initial burette reading was recorded. Next, 10 ml of orange juice was transferred into a 250 ml of conical flask using a pipette, and then 20 ml of distilled water was added to dilute the sample and make the titration process easier to observe. And after that, 2 to 3 drops of phenolphthalein indicator were added. The flask was placed beneath under the burette and NaO was added slowly while continuously swirling the flask to ensure proper mixing between the acid and the base. As the endpoint approached, the pink color produced by the indicator lasted longer, and the titration was stopped when a faint pale pink color remained in the solution for about 30 seconds. Lastly, the final burette reading was recorded and the experiment was repeated to obtain more reliable and consistent results. So as you can see on this part, these are the results obtained from the titration experiment. A rough titration was first carried out to estimate the endpoint and required 16.50 ml of NaO, and 2 additional titrations were then performed to obtain more accurate values. The volume of NaO used in trial 2 was 15.40 ml, while trial 3 required 15.50 ml. Since the difference between these values was only 0.10 ml, the results can be considered consistent and reliable. The average volume of NaO used was then calculated as 15.45 milliliters, which is equivalent to 0.01545 liter. The balanced chemical equation shown on the slide indicates that 1 mole of citric acid reacts with 3 moles of sodium hydroxide. This stoichiometric relationship was used to determine the amount of citric acid present in the orange juice sample. So now let's take a look at the calculation part. Using the average volume of NaO and its concentration of 0.050 molar, the number of moles of sodium hydroxide used was calculated to be 7.725 times 10 to the power of -4 moles. Based on the mole ratio from the balanced chemical equation earlier, the number of moles of citric acid was determined by dividing the moles of NaO by 3, giving it 2.575 times 10 to the power of negative 4 moles of citric acid. The molarity of citric acid in the orange juice was then calculated as 0.0258 molar. Using the molar mass of citric acid, which is 192 gram per mole, the mass of citric acid in the 10 milliliter sample was found to be approximately to 0.05 gram. Finally, the percentage concentration was calculated as 0.5% weight per volume. This means that every 100 milliliter of orange juice contains approximately of 0.5 gram of citric acid. And overall, the experiment successfully determined the citric acid content of the orange juice and demonstrated that acid-base titration is an effective method for analyzing the acidity of food and beverage samples. So now let's move on to the next part.
Because citric acid is weak acid, acid and base dissociation constant was used to calculate the pH of citric acid. First is initial pH of citric acid. For initial pH, H+ was calculated using equilibrium constant formula. Then the calculated value of H+ was substituted into pH formula. From the calculation, initial pH of citric acid was 2.65. Next is pH of citric acid after addition of 10 ml of sodium hydroxide. After addition of 10 ml of sodium hydroxide, it neutralizes a portion of acid into its conjugate base, creating a buffer solution because sodium hydroxide is strong base. So, Henderson-Hasselbalch equation was applied to calculate the pH. From the calculation, the pH of citric acid after addition of 10 ml of sodium hydroxide was calculated to be 5.94. Last is pH of citric acid at equivalent points. Because citric acid is weak acid, equivalent points occur when exactly 3 mole of sodium hydroxide have been added for every 1 mole of citric acid. At this point, all acidic protons have been stripped away, leaving an aqueous solution of the conjugate base, which is citric. Base dissociation constant was used to calculate this pH. From the calculation, the pH of citric acid at equivalent points is 9.08. Last but not least, from all the pH calculation, weak acid strong base titration curve was plotted as in the shown figure.
So, to wrap up our finding, let's walk through the core principle behind our results. From the balanced chemical equation, the stoichiometric ratio between the analyte, which is citric acid, and the titrant, which is NaO, is 1 to 3. The endpoint is reached when the phenolphthalein indicator changes the solution color into pale pink, indicating the presence of H+ ions that have been neutralized. The average volume of NaO used is a direct volumetric measure. The acid present that represents the exact amount of base needed to fulfill the 1 to 3 stoichiometric requirement for our 10 ml juice sample. And that's all for this part.
In conclusion, citric acid content in orange juice sample was successfully determined by direct acid-base titration with standardized 0.05 molar sodium hydroxide. Phenolphthalein was used as the indicator to identify the titration endpoint. Based on the titration result, the amount of citric acid present in 10 ml orange juice sample was calculated to be 2.575 times 10 to the power of negative 4, and the concentration of citric acid was determined to be 0.0258 molar, which corresponds to 0.5% weight over volume citric acid. From the titration also, it was estimated that the pH of citric acid at initial is 2.65, 5.94 after addition of 10 ml of sodium hydroxide, and 9.08 at equivalent points. Also, the volume of sodium hydroxide used to reach endpoint was 15.45 ml. Overall, the calculated citric acid content indicates that the orange juice sample has an acidity level within the expected range for orange juice, and the experiment demonstrated that volumetric analysis is an accurate and reliable method for determining citric acid content in fruit juice.